Okay, let's begin. Thanks very much for tuning in this week. We've got over 60 people now on the list for these drop-in sessions, and what we really want out of this is an opportunity for anybody who is an early adopter of using biology for their soils to ask questions, all kinds of technical questions. If you're making compost, if you're making Johnson's bioreactors, if you want to cut out agrochemicals, especially now with the fertilizer prices going up, then this is something that if we can answer the question, we will. And to give you a bit of a background of where our expertise starts and ends, we have concluded a validation trial this year on spring barley, which was a relative success, where we managed to, in the same field, go up to 88% of the conventional side of the field in terms of yield, and the control was down at 41%. So that gave us the impetus to keep going. And in terms of biology, we're working with Dr. Ingham's concepts, where she talks about having certain minimum values for the amounts of bacteria, fungi, protozoa, nematodes. And I'm really proud to say that the guys who are on this call, I'm going to introduce them. They have pushed the numbers in these compost bioincubators, as they've been called, to higher than I have ever seen, and I would have thought I would have seen. So we're quite capable of answering the questions of how to do it. And then in terms of answering the question of how this works in practice, we have got some experience now to be able to say, what we can do actually in terms of applying this to the field. What we believe that still needs to be done is a lot of in-field trials to answer some of the questions that we still see in front of us. And so we are completely anti-dogmatic. We treat the entire company and this whole approach to growing food as an experiment, because basically we only believe in what we see. And so we don't want to come in with a big amount of hype. We simply want to come out with information that's actually actionable. So that's basically our expertise. I'm just going to introduce quickly the three faces on one of the screens that you see. Left to right. There's Tom in the checkered shirt. There's Luca in the red one. And then the blue shirt, that's Adam. And these guys will give you detailed answers to your questions in terms of how to increase the populations of microbes in whatever mediums you're trying to grow them out. And then what happens with them once we actually start putting them out into the fields. So there's quite a lot of information in those three heads. There's a little bit in my head even. So we'll be able to answer all we can. So is there any burning questions right now, or can I show you a little fun thing that we've been preparing?
Go fun, Daniel.
Shoot the fun thing, Daniel.
Okay.
Yeah, start with that, yeah.
Okay, fine. So I'm going to share my screen. And the thing that I wanted to show you is that we have been taking in samples from yourselves, from people across the country, people who have been trying to make their own compost, make the Johnson's bioreactors, which are becoming more and more popular. I'll try to actually make these numbers bigger. And the reason that we've put this together is because we want to start a bit of a Top Gear style leaderboard, and show who is actually making something, and we'd like to basically improve on all of this and see who can get the best numbers. And we still don't know how we're going to judge this leaderboard overall. We need to have like a very simple way of sort of giving a number, you know, who's on top, who's sort of getting there. But I can discuss in general what we're looking for. And what we're looking for is for all of these fields to be green, first of all. And this is why I'm so happy to have Judith on the call, because Judith in her garden compost bin has, and this is this line over here, she basically produced, by chance, I can only assume, a compost that is green across the board. And this is not something that we see very often. It's actually something that is very rare. And this is something that Dr. Ingham would call biocomplete. So there is a relatively, you know, not low, but adequate number of bacteria. There's no actinobacteria, which means that it can go to sort of be used at the next levels of succession. The amount of fungi is very close to the bacteria, which means that because it's about 0.7, I think I've got the number here, 0.6. So it's actually something that Dr. Ingham would say that is approaching the sort of vegetable stage, the regular vegetables, actually, what vegetables would like in terms of fungal to bacterial biomass ratio. And then the protozoa have got a very respectable, about 450,000 per milliliter of compost, and then you've got a decent number of bacterial feeding nematodes, and everything else is clean. There's no indication of pathogens in here. So this is it, without actually having some sort of a number to put on this. To us, this is a perfect compost, basically. And so this is a very healthy material. This is something that you can extract the biology and use it, or just use the compost. So this is the sort of golden standard that we would want to be moving towards whenever people are sending us samples. If we see samples, we'd like that to happen. Anyways, Niels raised his hand. Go for it.
Yeah, I was just going to say, do you want to look at maybe freezing the top row and freezing the left two columns on your spreadsheet, so we can see what's what basically when you're scrolling around?
Ooh, freeze. Good. Here we go. That's all frozen now.
Yeah, awesome.
Like Disney. So in terms of other numbers, you'll find that sometimes we don't count bacteria. We do these days for all samples, but sometimes in the past we haven't counted bacteria because of the amount of it, and not seeing the amounts of other things that we wanted to see. However, basically there was in here, there's only 2 examples of just very, very highly bacterial compost, and the rest is actually relatively good. So the compost samples that we get don't have an issue with too high of a number of bacteria. It's all adequate. But soil samples that we get have nothing else but bacteria. And so where we want to get to is a situation where the soils have got these sort of numbers that we see in front of here, so that we have a decent but not over excessive amount of bacteria, and then all of the other elements, you know, in balance. So one of the challenges is going to be not only producing a good inoculum, but also making sure that these microbes survive in the soil. And so it starts with this good inoculum. And so anybody who is doing it on their own, you know, what we're trying to get to is basically a bit of a balance. So then in terms of nematodes, you know, they vary, but most of these composts have got some number of nematodes. In terms of protozoa, and this is interesting, all of the compost, apart from one, have got protozoa in it, and then there's only 2 samples in this list which are missing one of the 2 groups of protozoa. But also what's important to note is none of them actually have got any ciliates. And ciliates, show us anaerobic conditions potentially, or wet conditions at least. So in general, the compost samples that we get sent in are not actually bad at all. There could be improvement on the numbers, but in general, at least when it comes to protozoa, they're actually quite decent. So what we have done in the lab here is we have managed to drastically increase nematode populations and protozoa populations, and we also found ways to boost fungi. So we'll be able to answer questions on that. But I just wanted to I'm going to stop this share now unless you've got questions regarding these numbers.
Yeah,
Harry.
Judith's beautiful sample, would that be good enough to grow a crop straight into?
Judith's sample a crop into it. Yeah, I don't see a reason why not. It's just that it's a question of quantity. Obviously, I've got an ambition to. I've got an ambition, obviously, to look at hectares and hectares and hectares. So it's about repeating that on a larger scale. But yes.
On the strength of that, if that was a field sample, would you be able to drill straight into that, do you think?
Yes, why not?
Yep.
Absolutely. I'll be very happy. If we had that in that amount of microbes in the field, then yes.
Surely, sorry, raise my hand.
Yes, go for it.
Beauty's an eye of the beholder, isn't it? Because we've got very highly composted raised beds in my garden here. You can't grow carrots in it, but it grows amazing lettuce, for instance. So carrots don't want that level of they want a much sandier loam blend where they seek to go down to get the nutrients, to get a long root. So this is highly dependent on what you're going to grow, isn't it? Sort of supporting the same point.
The answer to that question was based on the biology itself. If we had the numbers basically of biology that's in that compost, then yes, I will be happy to see that in the soil actually, because that's the sort of thing that we need to have in the soil beside the roots of the crops that we've got to even have the start of the experiment really. And so it's still a challenge to get to numbers like that in the soil, and this is something that we need to put the work into.
Daniel, quick question.
Yes.
Sorry, just leading off from what Rob Ward just said. So, is it a plan to sort of try and create different inoculums for different crops, or in the long run, or how do you see this going?
There are certain things.
If you're saying that, for example, if you're saying that stuff, for example, in Judith's garden compost, which looks amazing, is maybe almost too rich for carrots in simple terms, if I've understood him correctly, then do you need to pare it back a bit and create something different for a different crop, or what's the deal?
No, this will be actually good for carrots, but it might not be good for brassicas, because there's too much fungi in this, for instance. And so then you actually want to have actinobacteria. Like you've got the second column here, you've got actinobacteria per milliliter. Actinobacteria actually outcompete fungi in some of these lower succession soils, and that allows the brassicas to still get all of their nutrients, but don't get attacked by mycorrhizal fungi or other fungi. So basically, this is where we're actually having less fungi is better.
Can I extend that then? What are the pros and cons? The different populations are on each other. I think you sort of said it, but is one going to actually take out the other because of its quantity of population?
It's a very dynamic system, and it's not something that we'll be able to control completely.
Right.
But if we want to have the ambition of having very, very diverse multiple species mix, but it's something that the plants that we put in should be able to control by putting out the exudates that select for the microbes that are better for them. So we want to start with a mix that will allow the plants to start selecting, And so, for instance, if there's if there are early succession plants like brassicas, then we may want to have fungi, but we may not want to have 0.6. We may want to be down to 0.3 or even less. But really, the actinobacteria is something that we will be actually quite happy with, because they just work very well. But then as we move up, and you're obviously, Rob, you work in mostly horticulture. That's your happy place. My happy place is arable, and Niels probably, I'm not sure if he's going to confirm that, but I think it's Pastures that Niels likes to work with.
And mixed, but yes.
But the thing is obviously that then in general these require different amounts of fungi to be able to for the system to work. When I say that, I will go back to my initial statement. This is something that Dr. Ingham says that works, right? This is something that research shows that works. And what's not happened in this country is actually people taking this material and being able to control these conditions in the soil to that extent yet in practice. And so this is why we're pressing for in-field trials for next year so hard, because we want to gather all that data and we need it in real world.
Okay, so we got a little bit of insight here for you as well. The target numbers for the compost and what's actually hitting the field, what's being taken on by the field, the survival rates of the groups that we're putting out there, they're all going to be slightly different depending on the field, the crop, the inoculum, right? So we throw shit at a wall and some of it sticks, and then we have to keep throwing that same target, those same target groups at that wall until everything sticks. And that might take some years, realistically. Might not all take straight away. But our research here has found that the bacterial feeding nematodes do take really fast, and so do the fungi. So this is good news for us, because that's going to help develop the system, develop the structure of the soils, and make it easier for stuff like flagellates to also take. And maybe the more nuanced, tiny things that require more soil pores, more surface area in the agricultural soils. So yeah, it's just a little nuance there, maybe.
And also to add on, it's important to note that a lot of the soils we're dealing with are, for the most part, fairly degraded and have little to no fungi, little to no nematodes. You might be lucky to find a few bacterial feeders. If there are any, it's usually root feeders. And in terms of flagellates and protozoa, there's zero pretty much most of the time. And so we aim for high numbers in our composts, our incubators. And what ends up hitting the field, because it's diluted, you end up spreading it over a large area. You keep on applying that over time until your samples of the field are hitting appropriate levels for the target crop. So,
and also, I mean, this is something obviously that has been true in the past, and this is something that we're 1 second, Rob. This is something that obviously we're trying to gather data here, and what we don't know, for instance, is what's stopping these microbes from establishing. What Adam has related to things that we have seen actually stick. We were very, very pleased to see our nematodes just over 2 weeks after application multiply in the agricultural soil over 5 hectares, and
by a factor of a few hundred times, it seemed.
So basically, it means that that it is possible to get them established, but we still don't know exactly which species, when, what soils, and what conditions. And so, for instance, we don't only work with microscopes. We have sent samples away to Granta Labs to get them tested, in a classical sense of testing. And tomorrow I'm going to collect some more samples and send it to ALS for pesticide screens, so we know everything that we can measure, what could be potentially preventing them from establishing. And we go out with a penetrometer, we try to collect all that data. And so this is the sort of work that needs to happen in our view. Sorry, Rob, go for it.
Well, I mean, I'm only sort of trying to take the conversation further because it's fascinating. Because clearly, that throwing it at the wall, quite rightly, it's a good analogy, isn't it? Is there any obvious things that work better than others, other than the fact that, let's say, specifically nematodes seem to be working better in that situation? I mean, for instance, would a cover crop, applying it with a cover crop or some sort of leaf canopy, or even or actually injected in the way it digests, is there other some things that you think that clearly are going to be helpful?
We'd like to actually test all of that. There's a lot of information coming, from other places that support that, and so just this was an interesting conversation with Dr. Ingham as well, because I mentioned to her Dr. Christine Jones, in Australia, talking about multiple cover crop, and then Dr. Ingham said, yes, we've had this conversation with Dr. Jones, ages ago, 20 years ago. So the thing is that the conversation is about having a diverse mix of species of plants to be able to support different microbes at different times.
That makes sense. Yeah. And the other thing we saw in Florida, we're doing some work with a biostimulant rather than inoculant or biomass, was the benefit of feeding them was far more significant than they imagined. So what they did is trials with no biomass and biostimulants, and there was a benefit to add in the biomass, but the impact of not having the inoculant, or whatever you want to call it, to feed them, that was a far bigger number. So in other words, seeding with the microbes and then feeding the microbes. The feeding of the microbes was proved to be in Florida soil, which is a bit like Blackpool Pleasure Beach, but with less organic matter. I mean, it's almost pure sand. So that the logic there is that there's very, very little organic matter, and despite that, the microbes got away quickly. So and they got amazing results in strawberries, particularly. So these are raised beds, drip irrigation. So they were able to put it through the drip system on a monthly basis. So that may be also a key factor. So I don't know, just our experience.
No, it's actually a great point, and this is something that we've been discussing here over the last 2 weeks, actually. That not only this microbial mix could be used as a sort of microfactory of micronutrients, but potentially could be used as an effective pump of nutrients. And so there's still a lot of nutrient sources that come, that should be part of the circular economy, for instance. And this is something that I would be happy to talk with you, Rob, more about of how this could be actually, implemented on a much larger scale. We also have got ideas, of producing other nutrients and actually having them, available onto the microbes at various stages to be able to put that into the plants.
Yeah, and then it's that ratio of nitrogen, or the impact of nitrogen on any additional nitrogen to put on conventional NP and K, how negative that was, even then, so the opposite effect on microbes. There's work being done by Essex Uni, potentially with a large farm nearby, that are looking at that. Mm-hmm. And seeing how we can manage that. And then the issue, if you're using a feed through the drip irrigation system, as in a biological feed, you can use that through the conventional system, but if you're treating it through drip, you've got to have a separate dosatron. So it may be better to could you inoculate the plants, or could you start the season with a dose of your concoction, almost like slow release sort of pack of useful stuff, that then the rest of it takes over with a feeding system for the microbes.
I mean, totally. This is something that that needs to be looked into. You know, as a whole system. You know, all of the pathways that we're being told, you know, from academia that are available to use. You know, this all comes down to basically a nutrient flow. And so we need to have a full understanding of, you know, what plants are getting, and is it actually the biology that's doing it, or, you know, have we simply put out some biology and it died and provided the nutrients? What are we actually, you know, what are we actually doing? So all of this information needs to be gathered, and it needs to be gathered in a field setting, in my opinion. Phil, you've got a hand up?
Hello. Yep. I was just wondering, obviously with Dr. Lane's method with supporting the soil biology and the soil food web and things like that, are we not only expecting that to work in a fully organic situation?
Sorry, is the question, can it work in other situation, or
well, is it not impossible really for it to work in a conventional situation, especially with sort of fungicide applications, things like that? Does that not, unless you're trying to replenish it every year, which I suppose means that you don't get that much traction, or am I misguided with that?
You know, it's a fantastic question, because it's hard to find people on the you know, around the planet who, you know, actual farmers who are adopters of this, who just use biology and nothing else. And if the Simon that's joined us is actually Simon Cowell, you know, Simon is a great example here in the UK. Where, good afternoon. Where basically there is a yearly application of compost, but it's part of a conventional system with glyphosate, with fungicides. I've also spoken with a farmer family in Canada on a much larger scale, and they also use this kind of system where basically they use biology as an addition to a normal conventional system. And so that allowed them to reduce their inputs quite considerably, but they still work conventionally because that's what they're comfortable with. So people have integrated this, you know, very successfully. Obviously, the thing that I've, you know, saw quite a while back is that it's not the farmer's role to convert their tractor into electric from diesel. It's the industry that's supposed to come up with a better product that's sustainable, rather than basically the society dropping this onto the farmer to figure this out. So this is why we started this as a company, to be able to figure out all these things and say, okay, well, can we make this into a fully sustainable 100% biology system that the farmer can simply buy into, and it could be economically viable, economically competitive with the current agrochemicals. So to answer your question, Phil, directly, people mix and match. They still use normal chemicals and still use biology, and that allows them to reduce their inputs. Tim Parton is another example in the UK. And what we're trying to do is we're trying to make an end-all be-all system which just works with biology, and we want all of the questions answered. They want the questions that we still don't have answers to.
And with the work we did in Florida, we saw a 75% reduction in fertilizer, NPK, and a 50% reduction in fungicide. So that's reduction, not a removal. And you could sort of celebrate that rather than chastise it, I think. So you know, imagine if we took 75% of the NPK out of the UK farming industry in arable and 50% of fungicide out. Let's not go into pesticides right now because there's pros, you know, there's other things there to talk about, but that would be an enormous carbon change. And talking to BioMe Maker, who are the DNA guys from originally Spain, but now in the US, they felt very strongly that it is a blend of I don't really know what to call them now, but it's conventional. What wasn't conventional is now conventional. It's a blend of organic and inorganic. supplement or additives, and that's what their view. But we could go a lot more. We could, if it was 75 biological and 25 non, then that actually would be fantastic result.
Question.
But
Sorry. After you, Rob. Sorry.
Well, it's just saying it's not. This is happening in scale in the strawberry industry in Florida already. It's not something that's been, trialed. This is hundreds of acres, and and they're doing it because it makes money. Bottom line.
Rob, are they making money because their inputs have gone down? Is that the reason why they're making money?
Yeah.
Yeah.
There's $1,000 an acre better off. But they've also seen a yield uplift of 18% and significant improvement in shelf life and improved bricks, so better fruit eating quality.
Yeah.
Same experience of blueberries, same experience of watermelon, and all kinds of crops like that. They're now trying with 40 different crops, including grass. Well, they call it sod out there. So it's
what company is that?
True Algae.
Oh, True Algae.
It's a biostimulant. It's not this solution. Basically, what we need is this. We need Daniel's range of biologicals mixed with, I believe, the feed, which is what that is. It's a food. It's a stimulant, not a. It's derived from algae. Yeah. So I think the two are together, and what I was trying to go at before already. You can accelerate the growth of microbes with this feed dramatically, and that's what they're achieving.
And are they still putting down compost, or they're not using compost at all?
These guys in Florida,
yeah,
they're just using they are in some cases using a microbial treatment, but they're just using normal feed through fertigation through the drip irrigation.
Okay.
Sorry, Nick.
Yeah, sorry, Daniel. Is there any concern that if you were to try and go into what Rob was mentioning about the let's say 75-25% mix of some sort, if we're trying to get people to, let's say, use your products and reduce NPK, which of course would be a great thing to do, is there any concern that adding stuff like NPK might impact your products? Might it damage it at all if you were trying to sort of blend it? Or
there's a certainty about that, actually, and there's enough studies to say that it affects biology and affects also how plants speak to biology. Yeah. So it's something that prevents, in my view, prevents farmers from actually moving away from agrochemicals altogether. But I'm actually just rushing forward with that statement, because realistically what needs to happen is we need to have a complete system which basically allows the farmers to be able not to have to mix and match. And we do have some things that we're going to keep to ourselves basically right now in terms of what we're planning to test out, to be able to complete any gaps that we see, potential gaps that we see in the biological mechanisms, to provide everything that the plants need, but still in a biological fashion, basically in a sustainable fashion.
Can I say that's absolutely exactly what the industry should do. The reality of that transition from where we are now to not doing it, other than farm over manure or distillate, to go to what you're talking about is very tough and challenging.
No, it's not. It's definitely not easy. And what I think the ultimate answer to Nick's question, it would be, what we're trying really to develop is a way for the farmer not to have to be spending the same amount of money with us every year. We want to basically help the farmers to get to the point where they've got this fully regenerative system, and we come in with crop protection and feeding rather than supplying the entire soil food web every year, which had been damaged by addition of other chemicals. So that's what we're trying to do. Obviously, Nick, you're thinking also of your potatoes, which is going to be a bit of a challenge for you to actually have a constant cover crop established. I'm just thinking of some other potential crops as well.
Yeah, that kind of stuff. I mean, a comment that was made today when I was discussing this with somebody was, that your soil is so sandy and it just loses everything straight away, allegedly. That applying something like this product that you guys are doing, it might just wash through and be lost within a year or two. And I didn't think that was the right way of looking at it. And actually, going back to what your team of three were there was saying, one of them, I can't remember, one of you guys was saying about you need to keep on applying this every year to slowly but surely build it up, and suddenly we'll get to the point where it can sustain itself going forward, or if that's the right word to use.
What the guys basically what they were referring to is the fact that the conditions for all of the microbes that we put out are not perfect.
Yeah.
If you imagine basically these microbes, like we look at ourselves, right, and we're this complex organism, and this complex organism can eat carrots and steak and bread all at the same time. Well, each one of these microbes is so small that it can eat just one type of food from one of these groups, basically, and nothing else. And even just for the fact that we put it out and it doesn't have that particular food in the soil, that's it, it's going to go. And then any other change of condition, that's it, it's going to go. We saw even yesterday, we did a small test on a slide where I left drops of a compost tea, which had a phenomenal amount of protozoa in it, to see if they're going to actually enclose into a cyst and survive the drying process. And today I threw it under the microscope and there's nothing there, basically. So we're basically facing a situation where we're going to be putting things out, and a lot of them will not be surviving. And this is what Adam was referring to in terms of the survival rates and actually building that up. In terms of the actual application, what happens with them, they don't really wash out because they don't have hands, but they have glues.
Yeah.
A lot of these sandy soils, or any of these soils that have been managed traditionally, they actually have a lot of bacteria, which also produce some glues, and so they attract each other. They basically use the bacteria as food. So if we have the correct species to eat the bacteria that are already in your soil, then that means that basically these guys are going to stick. And I think this is, Adam might confirm his own suspicions when I say this, but this is probably what happened with our nematodes. They had exactly the correct food out in that field.
And actually, like a web of fungi in the field will help keep the calcium around and keep some of the nutrients in the upper parts of the soil strata, which is really important, right? Sand without fungi is just going to leach calcium. So it kind of goes back to what I maybe sort of alluded to earlier, which is that I guess you kind of need to make a product from you guys needs to be, to a degree, maybe bespoke to what is actually here at the moment or is not here on my farm, for example. Yeah.
Okay, Judith has her hand up for a while.
Yeah, it's all, for me, it's like trying to transition from conventional chemical stuff on our grazing farm to something that's either a lot less chemical or no chemical in the end. But I'm struggling to get my head around how we actually do that. How do we, I mean, I need to, okay, I've made decent compost in the garden, but I'm going to need a hell of a lot more of that out in the field. And it's how do I actually control and have any knowledge as to how best to put that in place, especially now we're under this pressure with the fertilizer prices having gone wonky, and I don't see it come back.
How much fertilizer do you put on your pastures, if I may ask?
Well, I'll be honest, I have never really understood fertilizer, but my husband tells me it's 3 bags an acre. So is that 75 kilos an acre? Now that's a complete mismatch of terms to me, because there's conventional and it's kilos per acre, but that's roughly what on rough average
Mhm.
in the spring, and maybe a bit more later on if we feel we're short. But I mean, we looked at some of our soils, and I mean, there was, like you say, it was bacterial, no fungi whatsoever.
Mhm.
And there was a half decent, I think there was half decent carbon content there, 5 or between 4.5 and 5.5 or something.
Well, that's a start, because Adam, did you tell me about sort of like the minimum organic matter requirement for even to start thinking of adding the microbes? You're muted.
It's 2%, I think. That's the information out there anyway.
So, you know, if you have 4 and 5%, then already you've got something to start latching on. But obviously then your question is about, you know, how do you produce enough really? Obviously, you know, I can tell you, yes, of course.
What's the best way to apply it?
Say again, please.
What's the best way to produce it? And, you know, I can produce an amount, but I'm presuming that you would need to do some sort of a micro compost tea or something. And if you're doing that, depending on how you're feeding it, are you actually encouraging the growth and multiplication of bacteria? You're not going to multiply fungi in that, are you?
I'll let the guys answer that.
I don't think.
Probably not, no. We haven't seen it happen. So, like the beneficial soil fungi, anyway, the types that live in our soils and are probably usually tertiary decomposers. They live off metabolites of other decomposers in the system. So, no, they're probably not going to grow out. They're, you know, the secret ingredient for those guys is really time.
Yeah, so you're better just using. I mean, I haven't been able to actually get a Johnson Sioux started, but I'm better going with that and using that.
I mean, you can definitely start. There is no guarantee that Johnson Sioux is going to give you the fungal numbers, because we've had samples of Johnson Sioux, and then people would come back and say, "Is like, how is it that after 14 months I don't have the number of fungi that I was hoping for?" And just you don't have it.
And are you counting fungi like hyphae or the fungal spores? Because I'd have presumed in Johnson Sioux you'd end up with fungal spores, and that's what would then generate your fungi out in the field. Or am I completely off?
We don't count spores in this lab, and we are fully aware of, you know, Dr. Johnson's advice on counting spores, and you know, we've got the sheet that he produced, you know, of what the spores look like and whatnot. But the idea behind spores is that we don't know if they're going to wake up, when they're going to wake up. So the spores are still, you know, a good indication. So yes, if you can produce material that actually has a lot of spores in it, and if you, for instance, send us a sample to the lab and say count the spores, we'll count the spores for you, and we'll be able to see if there's a correlation between, you know, counting the spores and then seeing if there's a result in terms of actual growing hyphae in the soil. So the actual, you know, end result is in the soil, right? But what we work with is actual hyphae, and so what we always try to do in the lab is to grow out actual growing living hyphae in our compost, so that we can work with that directly.
Right. Okay. And if you're going that's what you're doing, applying the biology. How do you then, on the other side of the equation, reduce the chemicals? I presume you don't just stop it, because that's going to put the soil into complete spasms, isn't it?
Well, this is something, Niels is nodding his head. This is something that we don't want to be answering right now, because we haven't done enough trials. And so none of our trials are designed to go on full field scale or full farm scale at the moment. And we don't want to put that risk onto the farmers, but we need to still gather that data of what would happen when we actually apply just biology. And so far we know that on barley we were able to not get the same yield with, you know, not the best application of biology, but it was close. But we also showed that the control was very, very poor. So we knew that we were doing something, and now we're doing more with the winter wheat trial. So the idea behind what we're doing is to be able to answer that question, but we won't be able to really honestly answer that question until probably the end of next year. So if you're thinking of turning, your entire field, the risk to you and to us will be too great to even start that conversation. And I think what you could do is you could simply start applying this biology if you have good biology. If you have enough of the biology, I can tell you the application rates that we use. And then you could, based on the amount of material that you produce, you can then design your experiments to that scale and then see how much you can, get away of reducing your fertilizer. Because you might be able to see how your plants are behaving and then decide that, okay, I'm going to put on some fertilizer. I'm not going to leave that without any fertilizer. But then you'll be able to, at the end of that trial, you'll be able to measure how much did you actually have to put on. So don't ever go, head first, because it just might actually hurt you. So we don't want that to happen.
Yeah, you couldn't afford to do it in the whole place. You just pick a wee field somewhere, or a strip, or a corner, or a whatever.
No, not at this stage, no. And Niels have got some methods. I think he's waving his hand. I think you can do something, potentially.
How much have you made?
Oh, I mean, the biology that I'm talking about is just a wee pile in the garden. But we're going to need to make a whole lot more for, on a farm scale if we're going to make this change.
So what we're trying to do, obviously, Judith, is to be able to produce this for farmers. And at the moment, our prices are not competitive with chemicals because of our scale. So we can't do a economically competitive trial, but the actual aim is that, you don't actually have to produce it. But yes, if you were to produce it, how much acreage do you have?
That we actually spread fertilizer on?
Yes.
350, 400 acres of grassland.
Okay, so about 200. And we've been adding at each application, we've been adding about 70 liters of compost per hectare. So then, if you have this, 100,
0.70 liters per hectare.
Yes, yeah. Times 200. Yeah, so you basically need 14 cubic meters of compost per application for that 200 hectares. And we would, in the arable regime, we would be adding it 5 times in a year, minimum. And so then you're looking at 14 times 5, in a single year to be able to do this. So the amount of we're looking to find ways to concentrate this biology even further than what we've done so far to reduce these ratios, but we wouldn't go below that at the moment. And some people advise, as low as 2 pounds an acre, and we don't see that happening anytime soon.
You're doing horticulture.
Maybe.
No, I'm only kidding. Is there a way, if you dry it, you talked about assist mode, and how much can you make it dormant in a fully drying mode?
We don't know yet. We don't know yet, and I think this is something that that is an interesting challenge to us at the moment. At the moment, we've got to a point where we're capable of extracting the biology and storing it in cold storage and transporting it using chilled transport, but we still treat it as a perishable item. But Adam, you have done some experiments with trying to chemically put things to sleep. What was that like?
Yeah, it's all been pretty terrible so far. We've tried using sugar. A classic one is phosphoric acid, so slowly raising the pH to cause the microbes to insist, using sugar to dry out the liquid, so to speak. So you're binding every water molecule with a sugar molecule, so it becomes like a syrup. And my idea was if you did it slowly, maybe they would insist as they felt the drier conditions come into the environment. But nothing so far. Obviously in nature, stuff dries out. So maybe there's a rate at which you need to dehydrate the solution in order to get them to 100% of them to insist without mortality.
So most of the information that we've had, even from Dr. David Johnson, actually, the author of the Johnson Soup bioreactor, is that if you're using biology that has been dried, for instance, you take your seeds and you coat them and you dry them, that's less viable than fresh biology. That's why at the moment, because those systems are still, not 100% researched properly, we wouldn't want to be, trying to, we wouldn't want to lose viability because of, trying to transport it. Sorry, Phil had his hand up quite
Okay,
for a long time as well.
Hello, sorry, I had a phone call, so I apologize if this has been covered, but one thing, just let me know if it has, I'll stop talking. One thing we're struggling with on the farms, getting our fungal content higher. Obviously, well, we were a conventional farm turned organic 2 years ago, so we're just in our first year of organic this year. Obviously, fungicides are hugely effective in a conventional farming situation, but they'll kill beneficials and non-beneficial fungi. But what we're trying to do, and I was just wondering what your thoughts were on this. Obviously, making compost on site here to make compost teas and extracts, we're trying to inoculate our woody material with, I suppose, any fungus or anything that we can find, good fungi, basidiomycetes, things like that. Is that a good thing to do? Or, I mean, you see as well, some of the guys in the KNF world are sort of using IMOs and things and catching fungi, if you like, and doing it that way. I forget the guy's name, I'm sorry, that was talking about drying out biology. Certainly the KNF guys are using some of the sugar 50-50 ratios to try and do that to make it shelf stable. But I was just wondering what your thoughts were.
Shall I go for it? Yeah, so we've taken the IMO process here, and using microscopy, we've developed it. So we found a few things wrong with it. Possibly the visible molds you're seeing aren't true fungi and could be pathogenic. So IMO could be potentially really bad without the microscope, right? With the microscope, we found that you can drive it into a positive direction by manipulating it in certain ways. This could be by turning it to add aeration. It could be, I found, use an acidic high N input instead of an alkaline high N input. So instead of an oat flour, you'd use a coffee ground, and that drives the pH environment of the pile towards a woodland. So then you replace the omycetes, generally considered pathogenic fungi, or now in protozoa. It replaces them with beneficial fungi, and it replaces the ciliates with flagellates and arcella. So it starts to look a lot more healthy, and it starts to look a lot more like compost. So that's the developments we've made here. The IMO system's a really good system, but no one's ever tried to dial it in with the microscope. So it's quite exciting work, and it's part of the reason we've achieved such high nematode numbers in what I call like a mesophile expansion pile, something like that. We're focusing the mesophilic forms of life. We're focusing on them and trying to get them to replicate in huge numbers, which we've achieved. So yeah, and those piles are 16 to 22 days. So they're quite a fast pile, and they give you good bang for your buck.
Okay. Chris Trump, I know he's not local to us, seems to be doing a bit of a hybrid between Dr. Elaine's work and KNF.
Yeah, it's true, but
obviously not that relevant.
There still seems to be a lot of omycetes in the piles, which concerns me, right? Because we could focus on things we maybe need a lot more. Then again, the whole beneficial fungi omycetes conversation is quite a detailed one, and little is known about these organisms at the moment because of their huge diversity, right? Yeah, there's a lot of diversity out there. So it could be potentially, say you grow out lots of fungi that's eating wheat germ, and then you apply it to a freshly germinating wheat seed, You know, you can see where problems could occur. You just trained like type of fungi to eat wheat, and now you're applying it to like a germinating little fragile seed. It could actually increase damping off and other stuff like that, but we don't know right now. What we found out is, you know, that acidic high N input seems to be really cool.
Okay.
Seems to change the environment in that IMO pile, and the sugar and stuff, just leave it out. It's killing stuff. You want to completely skip IMO 2 and go to IMO 3 with a really as diverse inoculum as you can get. Whether that's good compost, biocomplete compost, great soil from a forest, or all of those things.
Because I mean, obviously with Dr. Elaine's sort of, say for example, 60% woody, 30% green and 10% high nitrogen for compost, we were trying to get the fungal content up in our woody material before we composted it, if you see what I mean, just to try and get a start. But I haven't actually used any IMOs yet, but my worry was that obviously the sugar in the IMO too, because if we drop them into a compost tea, then obviously that's just bacterial food, the sugar going in there.
Just don't waste your time on IMO 1 or 2 if you can. Just make as good compost as you can and use that as inoculum to go straight into IMO 3. And don't worry about greens in that pile. Just use high N and carbon and try and minimize the particle size as much as you can. So powders, meals, oak fines, you know, beech fines, stuff like this, really fine, like particles, not big chunks, and you're going to get much faster process. You're really looking in that process for nematodes and flagellates, right? The fungi part of that process is a bit of a con, maybe. Don't worry about that. Get a microscope, go for the nematodes and flagellates.
Okay, cool.
They're going to provide you with a decent amount of nutrient cycling. And they're going to take, well, the nematodes definitely going to take to the soil. They're going to maybe drive down the bacteria in the soils. This is what we hope. That's going to decrease the amount of alkaline glue in the soil. And then if we apply fungi at the same time, it's hopefully going to help those fungi grow out and start to prime the environment for their takeover. But that fungi you're seeing on IMO piles isn't necessarily beneficial soil fungi.
Yeah. Okay.
It might be good in the mulch layer, but we don't really have deep mulch layers in the field. So it's a lot up in the air at the moment. But definitely we've made a lot of progress on these kind of piles, and keep the sugar away from the IMO 1, or just don't use IMO 1. Use the best compost you can get, which is probably from us.
Cool. Thank you.
Slightly random question. What are they using in the human industry around probiotic tablets, particularly with slow release capsules? Is there anything there that we can learn from that could be useful here?
I mean, in terms of what, actually having the bacteria inert?
Well, the claim is that that's bacteria or beneficial bacteria or microbes in the tablets, but also the fact they're using slow release, different cellulose or different types of materials to stop it getting destroyed in our stomach or our small intestine, and then gradually be released later on when we need it. I just think it's the same principle in a field context.
Great question. It's the closest I've seen is somebody putting rhizobium into capsules. Can't remember which company it was, but basically saw something that somebody was who was producing rhizobium inoculum. They were putting them into little capsules like this, just like the gel-like capsules that you would swallow with your inoculum for the gut. But so that would ask maybe work in a similar fashion. I don't see necessarily a problem with drying out bacteria. We have an issue with drying out all the other bits. So bacteria don't seem to have, there's lots of them. And so even if they don't, if you have a high concentration of one species of bacteria, and 80% of them don't survive the drying process. You're going to have 20% feeding on the 80% that is still there as nutrients, and they can simply bounce back within a day. So I don't see an issue with that. We just have an issue basically with ensuring that all of the other bits of the soil food have actually survived. So that's the bit of a challenge. I saw Brian join us very, very late on, and this is actually something that an hour has already passed, and I didn't want to keep people. And suddenly this conversation is getting more interesting. Hello, Brian. You're right.
All good. Yourselves?
Very good, very good. Did you have any questions? Because obviously you popped in late.
No, as I say, it's a drop in, so I'm just waiting to see where the conversation's going at the moment.
Not a problem. So, at the moment, basically, I was about to start to wrap up. So so next week at 4:30. Hope to see you a bit earlier if you can make it, and then we'll be able to give you a bit more. Any more questions?
Just one question before I go. AMF, when that is dried out, I think in previous conversation you said that actually it was probably not viable. Is that still the thought?
That is the worry, because what we're talking about in most AMF products is just chopped up roots of plants that had been colonized.
Okay.
And so that has a shelf life, and their survivability, obviously, in that chopped up state is much lower than the survivability of a spore. And this goes back to Judith's questions. Judith has just disappeared, but actually finding an AMF product, Judith, we're talking spores right now.
Yeah.
Because Harry was asking about survivability of AMF products, arbuscular mycorrhizal fungi. The stuff that they tell you to add to your pasture as well. The thing about most products is they're simply chopped up roots of plants that had been colonized by these mycorrhizal fungi. And so then they're dried up, and they've got a shelf life, and it's really an unknown shelf life, realistically speaking, because it's very, very varied. And so what we really want is a product that has AMF fungi in a spore. And that is much more difficult to come by, because it's a much more difficult and demanding process to be able to get these spores. I think I've seen somebody in India producing it using aeroponics to be able to collect the spores, and it's a bit involved process. So there's not many people doing that. Most people just chuck the plants into soil that they found some mycorrhizals in and hope for the best.
Okay. Daniel, thank you very much for that. I need to go now, and it's been absolutely fascinating. Thank you very much.
Thank you for coming.
Yeah, well, thank you.
Well done. Great. Nice to meet you all.
Right, great. See you next week then. Anybody who wants to pop in, see you next week. Thank you very much for coming.
Hey, sorry.